Uh, Great. So thank you, everyone. Excited to share my sort of story and, intertwined in that the research that I've been doing since I was a graduate student, now a postdoc. And so the title of this talk is Why Do Particles Make People Sick? Using a multidisciplinary approach to understand particulate matter toxicity. So I was born in Monterrey, Nuevo León, Mexico, down here. So that's where my story starts. When I was a kid, we moved sort of, as Stephen said, far Texas, right here on the border. And I went there recently, and you can see this is kind of the entrance to the town. You get a picture of a cowboy waving. And then in my teenage years, moved to the southeast and went to high school in Pearland. Which Pearland High School, the only thing it's really known for outside of football is Megan Thee Stallion went there about a decade after I was there. So after moving to Southern California, I went to Moore Park where I did an associate's degree and transferred. And while I was an undergraduate doing chemistry, I did the physical chemistry concentration, which turned out to be really helpful in my graduate research. I got really involved in undergrad research, trying to figure out what I wanted to do, whether it's an industry trajectory or continuing to do research at master's or PhD level. So I actually started as a research assistant in a synthetic organic chemistry lab with Paula Diaconescu for about a year, which was really intense OChem synthesis, and after a while realized it wasn't really my cup of tea. So I made a transition to be an undergrad researcher with Suzanne Paulson in the Atmospheric and Oceanic Sciences Department, doing atmospheric chemistry. And I really loved that work so much that I applied to the atmospheric, or AOS for short, program, PhD program, and continued my research with Suzanne Paulson. And during that time, I was able to get some secondary mentoring from Andy Gio, who's a pulmonologist and toxicologist at the EPA. I'll tell that story a little bit more through the research. So after that, graduating, I started my postdoc with Dr. Jesus Rahu, who's a physician scientist and a pioneer in the field of environmental cardiology, trying to understand the interaction between the environment and health impacts. And so we've done a lot of work on air pollution and cardiovascular disease, as well as the emerging science of e-cig, vaping, health effects and things like that. So jumping into the actual research part and science, I just want to start with some definitions and primers before getting into everything. So when I say the word aerosol, very generally we're talking about a solid or liquid particle suspended in air. It's fine to think of it as spherical particles in this talk. You'll also hear particulate matter, or PM, which generally refers to aerosols associated with air pollution. So you'll hear me use those two words interchangeably to refer to air pollution particles. So local inflammation and systemic inflammation are two things that I want to point out ahead of time. When we talk about inflammation, we can think about it as an influx of white blood cells to a site of injury. And so, like you can imagine, if you prick yourself with, a piece of wood, it'll swell up and you'll get this local inflammation. When we talk about heart disease and systemic inflammation, it's the same process, but in the vascular system throughout our blood vessels. So it happens throughout the entire body. So we call that systemic inflammation. In terms of chemistry, we'll be talking about reactive oxygen species, which are these three in red here: superoxide, hydrogen peroxide, and hydroxyl radical. And we'll be talking about their kinetics. So we take a chemical reaction and we look at the rate, we can come up with an equation like this. We're going to be interested in the hydroxyl radical in this work because it's the most oxidizing of these species and can damage biomolecules. Short story there. One of the things that generates reactive oxygen species, free radicals, turns out to be iron. So we need to talk a little bit about iron chemistry and iron chelators. So in the inorganic form, in aqueous system, which is going to be the phase of chemistry we're doing in this talk, these tend to form these radicals, these ROS, slowly in solution. But there are other molecules that can chelate or bind iron and form these complexes. An example that will be important is citric acid, which can bind to the iron 2+ atom and form these kind of complexes here. And it turns out this is important because these complexes can have dramatically different properties than just straight inorganic iron. So for example, they can speed up or slow down this ROS generation, and they can enhance the solubility of mineral forms of iron. So from a chemical perspective, we want to know about ROS and iron. Kind of getting away from the molecular level, let's talk about air pollution, which if you've been in any urban environment, you maybe have seen something similar to this. So I'm going to describe the terrible flavors of air pollution, which are two main components: gases, which I've listed here, which won't be so important for this talk. The second component is particulate matter, aerosols. And to get an idea of the scale of what we're the size of what we're talking about here, this picture shows a human hair, and then these red particles are what we call PM 2.5 particles, which means they're particles that are 2.5 microns smaller. And one of them is laid on a PM 10 particle here in blue, which means particles 10 microns and smaller. So you can see how tiny these particles really are compared to the human hair or sand. And it turns out these are regulated in the United States by the Clean Air Act. So there are limits the government puts on the concentrations of this stuff in the air. And recently, ultrafine particles, diameters less than 0.1, become of interest because of their suspected toxicity. So very briefly, how do we get aerosols? How do we get these particles in our atmosphere? I want to describe what we call anthropogenic sources, which means human sources. So for example, biomass burning, fossil fuel burning is something you'd probably expect. Human-caused wildfires, transportation. But one thing I want to point to here on the bottom, because it's very interesting chemistry, is consumer products. So the volatile organic compounds, or VOCs, that come out of nail polish, wood finish that you keep in your garage, make it into the atmosphere as gases, and do some very interesting chemistry that was actually discovered in LA. And one of the things that Los Angeles is famous for is the smog. And so if you take a Google for smog pictures of Los Angeles, the 40s and 50s, you'll see some really dramatic pictures. And this was the first smog episode in LA back in 1943. There's a bit of a mystery of what was causing this intense smog, because up until that point, it was thought that coal burning was the culprit, and there wasn't any of that going on in LA at the time. So in the late 1940s and early 50s, Professor Ari Hagensmith from Caltech did some really interesting chemistry experiments. He built something called the smog chamber, just a plexiglass chamber with some plants inside. And it's a bright Southern California, sunny day. And he hooked up the emissions, or the tailpipe of a car into this chamber. And he was able to, concentrate the light, sunlight onto this from the exhaust. And he found that only when the sunlight was there did you get this brown formation of smog. And so what's the chemistry here? This is called photochemical smog. One of the ways you get air pollution in urban areas, another term for secondary organic aerosols. And basically, when you have emissions of nitric oxides and VOCs from vehicles or other sources, it can react in the sunlight to form ozone gas, particulate matter, and other toxic organic compounds. And in places like Los Angeles, you have something called a temperature inversion, which kind of keeps a cap on the local atmosphere. And this is responsible for the classic smoggy LA image. And so this is an important process, very chemically interesting, that I wanted to kind of bring your attention to. And it's going to be relevant to what we talk about in terms of toxicology. So zooming out again, I want to sort of describe the large big picture problem of air pollution health effects. So you probably don't need to be a scientist to know that inhaling stuff from like wildfires and cigarette smoke causes certain health effects. But epidemiologists are the ones that do studies that tell us what these particular health effects are and what they're associated with, for example, heart disease, lung disease, etc. What it doesn't give us is causality. Why do these particles, why does this pollution cause these health effects? And so perhaps that answer can lie within the science of aerosols and the chemistry of these particles. So, for example, in wildfires and cigarette smoke, we're interested in two chemical components, something called fulvic acid and humic-like substances, and iron. And we're interested in these compounds in the particles because they have an ability to generate reactive oxygen species. And it turns out these substances and this chemistry pops up everywhere in AOS, like cloud water chemistry, the chemistry of the ocean, rivers, things like that. But it turns out to be important in the lungs as well. So perhaps through this kind of understanding fundamental chemistry of these things, we can have a link to biomedical sciences and toxicology to find causality of why these particles, inhaling them, causes these effects. And so the broad research goal of my work is to understand the relationship between the chemistry of the particles and the biological mechanisms understanding particulate matter health effects. Or in other words, why do particles make people sick? Okay, so recently it's been appreciated that particulate matter inhalation, or air pollution inhalation, impacts every organ in the body. But we see the biggest health effects from the cardiovascular system. So it's been estimated up to 80% of premature deaths from air pollution exposure are due to cardiovascular disease, which is a surprise considering it enters your lungs. And out of all the components of air pollution, it turns out that these particles, the aerosols, are the components most strongly linked to health effects. And so why is that the case? What makes these particles, particularly the PM2.5, which are the most strongly linked, what makes them toxic? So there's two things that we know about particles that contributes to their toxicity, and that's their size. So if you think about a human lung, and you're breathing in smoke or you're breathing in pollution, the smaller sized particles are going to be able to penetrate deeper into the lung. So the PM2.5 particles make it deep into the lung where there's not a clearance mechanism, and so they're able to deposit there. So that's one reason. The other reason is dose, essentially. So if you have more smoke in the air, you breathe more of it in. So those are the two things that we know contribute to the toxicity of these air pollution particles. But there's still a lot of mysteries when it comes to the chemistry, like particle composition. It turns out, depending on where you're at, where the particles come from, the chemistry is going to be wildly different. So without getting into any of the specifics, these two pie charts, this is the composition profile of an open biomass burning particle, think a wildfire particle, versus a diesel exhaust particle. So think of a car exhaust particle. And just looking at the two colors, you can see that there's differences in the composition, which potentially is going to influence their toxicity. So when we ask this question, why do particles make people sick? Two of the things we don't know is how components induce toxicity. In other words, the biology of what's happening from aerosol inhalation to health effect. And the second thing we don't know is what makes the particles so toxic. In other words, the chemicals and the particulate matter that are responsible for toxicity. These are two pressing questions. So one hypothesis from sort of a biological perspective of the health effects has to do with reactive oxygen species and inflammation. So there's this idea of oxidative stress, which is the overproduction of reactive oxygen species in biological systems. This leads to damage of biomolecules. And so it's thought that this induction of oxidative stress is thought to be a key step in the onset of inflammation. And as it turns out, every health endpoint associated with particle inhalation involves inflammation. So potentially there's a link between the chemistry of the particles and their ability to generate this stuff and the inflammatory health effects we see. So the idea is basically if you have a balance of antioxidants and free radicals in a biological system, you could potentially have particle exposure or a cellular response to inhalation of particles that tips this balance and overwhelms your antioxidant defense system, manifesting as inflammation or tissue injury. So it's a very sort of simple explanation of it, and it's not the only hypothesis of how inflammation or health effects from particles arise. I just wanted to say that out front, but this is sort of the oxidative stress paradigm. So when it comes to the chemicals that makes particles so toxic, there are some candidates that pop up in epidemiology studies. Two of them that we'll be focusing on here are iron, so redox active transition metal, and some water-soluble organic carbon, which is something we call HULAs, humic-like substances, which I will explain in a bit. So these particles may be toxic, or these components of particles may be toxic because of their ability to generate these ROS. So if I have to tell you about humic-like substances, I think first I should explain what humic substances are. And the short story is both of these organic substances are water soluble, and they chelate, or they bind iron, and they speed up ROS generation. Okay. So we'll start with humic substances, which you find in the soil. And these have been studied for a couple hundred years. You find them in aquatic and terrestrial systems. There's extensive data and standards you can find and buy. So there's two fractions, a fulvic acid fraction and a humic acid fraction. And then we have HULAs, humic-like substances, which are very similar chemically to these soil substances, but they come from burning plant matter. So you find them in wildfire, wood smoke, cigarette smoke. So here are some molecular weights if you're interested in the size. Just to say that the stuff you get from burning wood or cigarette smoke is much smaller chemically, about 100 grams per mole, compared to the stuff you find in the soils. Right? So if you wanted to look at a chemical structure, you could think of this as sort of a subunit of larger molecules that make up these substances. It turns out that both the stuff from the soil, the fulvic acid, and this HULAS material are suspected to be components that contribute to the toxicity of wildfire and cigarette smoke in the lungs. And it turns out that this ROS chemistry of both of these substances binding with iron, their physiological conditions is not well known, although this stuff has been studied in the environment to some degree. So what we're going to do in the work I will show is we're going to take the stuff from the soil, fulvic acid, and because it resembles chemically the stuff we find in the air, HULAS, we're going to use it as a surrogate for the atmospheric stuff, because this is really difficult to come by. And what we're going to do is we're going to look at how hydroxyl radicals, these OH dot radicals, are generated by the binding of these organic substances with iron in the environment of simulated and human lung fluids. This is in the context of health. And it's going to be combining experiments with a little bit of modeling. And just to drive home the point that studying components of wildfire and cigarette smoke are important, as the United States and the world has made progress on improving air quality and due to vehicle emissions. For example, look at LA between the 60s and beginning of lockdowns in 2020. A lot of that work is being stifled by the increase in wildfires that's happening due to climate change. So just a few months after a relatively clean compared to decades before, a wildfire in Southern California just diminished it to the worst we've seen in decades. So there's still a lot of work to be done here. And refining the question even more now is, why do wildfire particles make people sick? Is really what we focus on. So what makes them toxic? Potentially this organic substance called HULAS. And how does it induce toxicity? There's some binding between this material and iron, and it generates ROS, leading to biological effects. Okay. So moving into the actual work briefly, I will just talk a little bit about the chemistry and the science of what I was just talking about, and then briefly talk about my work in the environmental cardiology lab that's been going on recently. So this is the alveoli connected to the bloodstream in the lungs. Okay, and so we're going to have a guide for these two parts. Since we're talking about wildfires, we'll have Willie the wildfire particle kind of guide us through the passage of the lungs and how we think these particles get here. So first, as you inhale, the particles come and interact with the lung lining fluid, which is attached to some lung cells here. Okay. So we're going to ask a question. Does fulvic acid, or the surrogate for HULAS, enhance OH generation from iron and lung lining fluid? And the hypothesis is this stuff strongly is going to bind iron and increase the redox activity, resulting in more radicals in the So we are going to use an experimental method. It's quite simple. We take a molecule called terephthalate, which can react with this radical here and form this compound. So it just adds this OH to this ring, and this stuff is fluorescent. So we're able to measure its generation in liquid. If we're careful about our calculations, we actually can calculate how many hydroxyl radicals are produced over some time scale, over some time span. So this is a very powerful and simple tool to accurately and specifically quantify radicals, which can be difficult due to their short-lived nature. For example, the hydroxyl radicals are around for about a microsecond, and then it reacts with something. So this has been a nice tool to investigate this chemistry. And we're interested in this chemistry of iron, right? So how does iron generate these OH radicals in the lung fluid? Well, it acts as iron in the +2 state acts as an electron donor to oxygen to form superoxide. And this is those reactions that I was showing earlier, and further donation of electrons forms hydrogen peroxide. And then this reaction here of iron +2 with hydrogen peroxide, forming the hydroxyl radical, some other products, is called the Fenton reaction. And it turns out in the lungs, we have some vitamin C or ascorbic acid present that acts as a reducing agent of iron 3. So pops this back into the iron 2 form, keeping this cycle going catalytically. So it turns out that this process is slow just for inorganic iron in lung fluid. But organic compounds, such as citric acid and humus, can bind iron and change its activity. And that's one of the things we're going to be probing in the context of the lungs. So first we want to simulate lung fluid, right? So we have two ways to do that. We have a laboratory prepared simulated lung fluid, which is just a phosphate buffered saline solution with 3 antioxidants that we find in the lung at concentrations we see in humans, sort of the average. We add citric acid, which is meant to mimic proteins that you find in the lung. Second, we have actual lung fluid that's collected from humans that we get from our collaborator at the EPA. And we call those bronchoalveolar lavage fluids, or BALF. And this is mostly salt and some lipids and proteins. We don't need to get into that too much. But the idea is that we have these model lung fluids, and we're able to extract or put chemicals that we're interested in from the air and use it as to mimic the environment. So for example, we'll be putting fulvic acid and iron into the lung fluid, and both of these investigate its chemistry. So remember, our question is, how does this interaction of the iron and fulvic acid generate OH? So we'll do this, we'll add it to our simulated lung fluid. We're going to measure OH radicals with the method I mentioned earlier, and then we're going to do 2 different types of modeling: thermodynamic and chemical kinetics modeling, basically to get an idea of what compounds are binding to iron at equilibrium. And the kinetics modeling will give us rate constants for ROS reactions that are important. What are our results? From the thermodynamic modeling, if you throw in this fulvic acid stuff that's a mimic for wildfire smoke into simulated lung fluid, it turns out it sort of plays a tug of war for whatever iron's present. Citric acid. What do I mean? So from our modeling, if we were to put a micromolar of iron 2 in the simulated lung fluid without any fulvic acid, it turns out most of that will be bound to citric acid, or the citric acid molecule will form this complex, with the rest of iron being in some inorganic form. But as soon as we run this simulation with the addition of some fulvic acid, you see here in the yellow that 94% of the iron is bound up now to the fulvic acid. The green here, which is the citric acid form of iron, only 2%. So as soon as we add a little bit of our fulvic acid, the iron shifts to be binding with this fulvic acid, even more so 100% when you increase it. So that gives us a piece of evidence that indeed this material is able to take iron away from citric acid. Does it make more radicals? Does it speed up this chemistry? So then we did chemical kinetics modeling, mixing experiments with the model. And without anything bound to iron, you find the rate constants for these 2 reactions, which turn out to be the 2 most important in terms of the model. And we were able to find rate constants for these analogous reactions, but iron bound to citric acid. And we found out that it increases it by 3 times here for this reaction and 76 times for the reaction that forms OH. So citric acid, organic molecule, binds iron, speeds up the rate constants of these reactions. Now, when we do that same modeling for the fulvic acid substance, which mimics wildfire smoke, we see an even higher increase in the rate constants of these reactions. So indeed, not only does this fulvic acid substance, not only is it better at pulling iron away from citric acid, it also increases radical generation to a larger degree. And so this was work that we published in Environmental Science and Technology in 2017. My first paper from grad school, so it's always holds a special place, right? So this is so kind of building on this. How realistic is this simulated lung fluid that we could just make in the lab? And we use different fluids that represent this or the human condition, and the answer is yes. So we're going to use the human lung fluid, and the way I got this was through networking at a conference where. When I was a nervous grad student presenting this work in Berkeley, I spoke with an analytical chemist from the EPA who said there was very interesting work that we were doing similar to the EPA. And he connected me to Andy Gio, who later would go to be a mentor. And it turned out his early work motivated the experiments and the work that I just showed. So it was kind of a nice connection to make. And also for me, a very clear point of the importance of networking and trying to meet people, particularly at conferences, that could be mutually beneficial and push your research to a different direction. So for example, now that I had the human lung fluid, we were able to get a little bit into biochemistry. And so I just showed some work in simulated lung fluid with the citric acid molecule that I said speeds up the free radical generation from iron. If you look in the biology literature, the proteins that are actually in the lung are albumin and transferrin, and they're supposed to do the opposite of that, right? So what's going on here? So it turns out, looking through the literature, nobody really had investigated how these proteins you actually find in the lung generate these radicals from iron. And so now with the tools that we had, and I'm not going to go through this data in the interest of time, we set up some experiments where we repeated these fulvic acid iron experiments in human lung fluid, compared it to what we call an idealized human lung fluid, where we just added the components that we thought were important, like albumin and transferrin, since they bind to iron. And we were actually able to estimate some rate constants that are relevant to the lung fluid. And so, for example, just to show the data, using what's called a rate limiting approximation, we were able to compare experimental data here in the blue with a model that we came up with using kinetics. And so that's this equation here. And after estimating rate constants for these reactions, we were able to plug that into our model. And you see the results here in the red. So we were able to get really good agreement with the experimental data that we measured in the lab, and then the kinetic equation that we developed, actually pen and paper, there's no computer in this case, which was kind of fun. And it gave us validation that these rate constants were indeed valid. And so this rate constant for albumin and iron reacting with oxygen, it turned out to be the first measure of that rate constant that had been reported. So we're very excited about that. That sort of ends the research part. And I know I have a couple minutes here. So just to get back to this question, why do wildfire particles make people sick? What makes them so toxic? Potentially this hueless material that we were using, fulvic acid as a substitute for. How do they induce toxicity? Maybe part of it is this hueless from wildfire, the cigarette smoke enters the lungs, where it chelates iron and promotes these free radicals from being generated, and that leads later on to inflammation. But one step, much more work needs to be done on that, more on an in vivo level. And so I will skip through this just to briefly talk about some of the work we do in the environmental cardiology and vascular biology lab, where basically we use mouse models, genetically altered mice, to understand disease progression of cardiovascular disease and the impact of air pollution exposure. So an early study found that mice that were exposed to filtered air versus particles develop more plaque in their aortas here. So this is a measure of plaque in the aorta, and the mice fed the same diet, exposed to pollution, they had more plaque buildup in their arteries, which was surprising. And so we're basically trying to dissect why that happens. Why does inhaling particulate matter cause plaque buildup or exacerbates that? And we think there's potentially a link with the digestive system, because we've seen inflammation in the intestines with exposure to particulate matter as well. And so potentially from the inhalation to the gut and gut bacteria and all this kind of biology, there might be a link to this plaque buildup. And so we're trying to dissect that using mouse models. And so we have exposure chambers where we're able to do experiments, in vivo experiments, and apply some of the chemical tools I was explaining earlier to more biology systems. And so, for example, we have a team of people that does everything from sequencing gut bacteria to imaging the plaque in the aorta, looking at lipids in the lipid panel and lung fluid, plasma, liver, white blood cell biology, PCR, and what's interesting, what I focus on is collecting the lung fluid, that I once got from someone else. And so now we're able to combine interdisciplinary methods to answer some pressing questions in biomedical sciences. And finally, just to say that we also work on electronic cigarette aerosol because of its increase in use lately, especially among youth, and basically a lack of understanding of the chemistry of what's in it. And it's very complicated chemistry that I won't go through, but it's still a big point of sort of black box information of how this material in your e-cig fluid vaporizes and heats up and generates toxic compounds. So very interesting organic chemistry where anyone's interested. So with that, I think I will end my talk by just putting the slide back up and just looking at the interdisciplinary nature of aerosol toxicology. And with that, I'm happy to take any questions that anyone has. Thank you very much.
Awesome. Thank you for sharing your story and about your research and everything. Steven actually had to go, but he did leave a couple questions. One of the questions was, how was your experience transferring into UCLA, and what were the main challenges when you arrived?
Yeah, it's a good question. I don't think I was prepared for the culture shock that I had going into a university like that. I'd grown up in Texas, and I'd been living in Southern California for 3 years and hadn't really adjusted yet to Southern California. So one of the big things was, it was a culture shock for me that I wasn't prepared for. And I found that definitely impacted how I went about my academics. And so one of the things that that helped me in that is to try to find some sense of community when I got to UCLA, and it was able to help me kind of progress and be a source of support as I got through the tough parts of exams and everything else.
A second question is, how did you get into research, and how did you overcome the struggles within research?
Good question. Yeah. So one of my chemistry professors, my first quarter when I transferred to UCLA, basically encouraged everyone to consider research as an internship opportunity as an undergrad or as a career. And honestly, up to that point, I didn't realize that scientific research was a thing. It hadn't really been, I just didn't know. And so he kind of opened that box for me by saying this is a thing, and gave us information of how you get involved. And so my very first research position, I got my CV together, what it was at the time, and just emailed it to a bunch of professors asking, hi, my name is David, I'm interested in doing research, here's my CV, let me know if you'd like to talk more. And eventually, Professor Deaconescu responded and said, yeah, we have a position open, there's room, why don't you come by? And, and that was it. So that was sort of the way I went about it. Even after I left that lab, I wanted to still get into research, sort of sent out emails more directed to a different field that I was getting interested in.
Thank you. I actually have a question about your research too, because
Sure.
I've done like a little bit of research on like aerosols, things like that. So I was kind of like, and I was wondering, have you guys used like bovine or porcine like fluid? Because I know like in the presentation you guys said that you used like a model. So I was wondering if it's like it was like made like artificially made, or was it like from another species?
Oh, good question. Yeah. So let me get that portion back up here. Yeah. So it's a very a good question. So with simulated lung fluid, this sort of formulation, it just turns out to be a very common one that was used in the atmospheric chemistry field.
Okay.
And there is basically no biological, material that's not a small organic. So there's nothing like a serum or that you would add, or even lipids, that you would find in the lungs. When we got the stuff collected from humans, we don't really have control over how much is in there, but we at least get what's there, what's actually there.
Yeah.
And there's been a lot of work of people that have tried to build a more realistic simulated lung fluid using the things that you've mentioned, and adding various lipids, vitamin E is another component. And it turns out to be pretty difficult, but it's actually used in cell culture applications. Now that I'm thinking, yeah.
Awesome, thank you.
Of course.
I guess I have a quick question. How did you guys determine like the exact chemicals in the smoke itself that cause these damaging effects?
Oh, very good question. So it's sort of like I don't want to say chain of command, but maybe a flow of information. So epidemiologists do these studies, data scientists essentially, where they look at, put it very crudely, correlations between particles in the air and health impacts wherever those particles are and people are being exposed. So that's how they did sort of the early studies. One of the first ones is called the Harvard 6-city study. So further studies, what they did is they didn't just look at the concentration of particles in the air as it's correlated to health effects. They looked at components of what was in those particles as they're linked to health effects. And so that's where some of these came about. So for example, before we really classified these particles based on their size, it was just total suspended particulate matter that was correlated to health effects. Later on, as we were able to measure and sort of distinguish between 0.5 micron sized particles versus PM10, and put that sort of distinction into these correlations, it was found that the PM2.5, the smaller particles, were most strongly linked to health effects. So it's sort of as the environmental science and the chemistry, the analytical chemistry of understanding what's in the air gets better, that information gets fed to epidemiologists that can see whether it pops up in the correlations. And then toxicologists can go and try to figure out the mechanism in between. Did that answer your question?
Yeah, I think that makes sense. So it's essentially working with other fields to gain like the data, and then you narrow it down. That makes sense, because it seems like there'd be a lot of stuff to work with, like when the raw smoke comes in to analyze that.
Yeah, and the other point about that is that these particles can have hundreds of thousands of different compounds at various concentrations, especially the organic components. So it becomes very difficult to tease out every single compound that's there, and so the field generally has tended to look for the ones that are most abundant and have a known or suspected toxicity profile.
Yeah, another question too for me.
Sure.
Do you like how do you guys saying that you guys are going into in vivo like experiments? Like how do you guys generate, like do you guys expose them to that 2.5, and how is that generated, like like those particles?
Sure. Yeah. That is actually a very interesting question. It gets into engineering a little bit, and so we have to call on our engineering friends. So there's multiple ways to do it. So for example, the chambers we have here, if you see this sort of orange stem going down, there's a solution there. And so basically what that solution is somebody went out with these high volume air pumps and just sucked the particles onto a filter. And so that's sort of filtration collection. And then they extracted it in water using like a sonicator and stuff, and it creates what they call like a slurry. So you take the particles from the air, put them in water, and then you put them here. This is called the nebulizer. And they have this system of flows that passes that slurry of particles in water and dissolved organics, what have you, through a desiccant here, which dries out the water. And then it goes through this system, which is a charge neutralizer. So it turns out aerosols are generally charged. And depending on the flow rate, it changes the sizes of the particles. It's kind of how the physics works out. And then it's able to deliver directly into the chamber. So that's one method. Other methods are, just having flow systems that take either ambient air or some type of, combustion system and then transport it into that. And it can get very tricky depending on, you want to control how much you deliver and make sure that's consistent. So that's why our friends at USC School of Engineering and the CIOTAS group have been, they developed this and sort of help us troubleshoot that.
Awesome, thank you.
Great.
Any other questions? Well, I just wanted to thank you once again for just sharing your story and sharing your research. It was a really interesting presentation, and it was really interesting overall, though. So I just wanted to thank you once again for it. So
I appreciate it, and welcome to provide my email if anybody has other follow-up questions. I'm happy to talk more.
awesome then. Thank you. Have a good one, though.
Thank you. Bye-bye.